Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of members of Cambridge Neuroscience. Cambridge Neuroscience is currently going through quite a detailed consultation process to develop 6 new themes for the research we do here. Each of the next 12 talks will come from one of the 6 new themes, 2 from each. For more info on the themes and the talks covered in this series, please follow the links below and follow us on Twitter at CamNeuro. This week we are delighted to welcome a relative Cambridge newbie, Dr. Jing. Jing is a group leader at the MRC Laboratory of Molecular Biology, where her team is working on revealing the developmental assembly of the serotonin system. Today, Jing is going to speak to us about the organization of the midbrain serotonin system. Welcome, Jing.
Thank you very much for the nice introduction, and it's my great honor to give this talk in Cambridge Neuroscience. Today, my topic is the organization of midbrain serotonin system. From the global mental health perspective, mental disorders like depression and anxiety disorders are on the rise in the last decade. Depression has become the leading cause of disability worldwide, and nearly 1 out of 5 people suffered from mental disorders last year in the US and UK. So the best weapons so far we have are the antidepressants to beat these mental disorders. And there are 1 out of 10 adults in the UK taking antidepressants. The number is 1 out of 6 for Americans. SSRIs, which is short for selective serotonin reuptake inhibitors, are the most frequently prescribed antidepressants. SSRIs lift up the mood by increasing the serotonin concentration in the synapse gap. At the serotonin terminals, there are serotonin transporters that reuptake the serotonin from the synaptic cleft back to the cell. And SSRI's function is to block the transporter, causing the actual cellular concentration of serotonin increased because of accumulation. The first SSRIs was launched at the market 30 years ago. So if increased serotonin can lift up our mood, does it mean that serotonin is in charge of happiness? I'm afraid that the answer is much more complicated than that. Serotonin is synthesized at the soma, and the key enzyme is TPH2, which is also the marker for serotonin neurons. There are 15 different kinds of serotonin transporters. Some of the receptors can located at the presynaptic sites, and some of them are postsynaptic located. In addition, some of the receptors are inhibitory, and some of them are excitatory. So the whole serotonin receptor system is very complicated, and it is impossible to deduce whether increased serotonin concentration is causing inhibition or activation at a specific targeting site. More than that, serotonin system innervates the entire brain. The dorsal raphe nucleus and the medial raphe nucleus provide serotonin to the forebrain. And DR contains the largest number of serotonin neurons, and it is the predominant source of serotonin in the forebrain. Here, you are looking at the impressive axonal projections of the DR serotonin system. This is the horizontal view of a mouse brain, and the olfactory bulb is facing down. The green structures represents the DR. So the DR serotonin system has extremely extensive axonal projections that are deeply engaged in the entire forebrain. Meanwhile, DR serotonin system is involved in multiple behavior and physiological functions, including anxiety, depression, sleep, memory, aggression, impulsivity, feeding, reward, and so on. So what is the relationship between the extensive projections and the wide spectrum of the functions? In other words, how is the DR serotonin system organized to manage so many outputs and functions? There could be 4 models. The first one, all the serotonin neurons in the DR is homogeneous. They targeted the downstreams randomly and collectively they tiled the entire target regions to create general serotonin levels. And in the second model, there may be functionally defined subpopulations, perhaps due to their different inputs, but each subpopulation tiled the entire target brain regions to achieve a similar outcome as model 1 we mentioned. And in the third model, there may be projection-defined subpopulations, perhaps as a developmental strategy to ensure that all targets are covered, but they are functionally equivalent. And then in the last model, the DR serotonin neurons are functionally heterogeneous, and their functions are defined by their projection size. So to find out which model is correct, I decided to dissect the DR serotonin system based on the projection pattern. First, I did a systematic retrograde tracing study. I chose 8 brain regions that are known for receiving input from the DR, and there are 4 cortical regions and 4 subcortical regions. I injected retrograde Cree to the targeting regions of a Cree reporter mouse line IF14, and the Cree traveled back to the DR and turned the reporter expression. After that, I collected brain slices containing DR and stained them with the antibody against the serotonin marker TPH2. And then I was facing the same problem previous study has ignored, that is the standardized registration of these results. So my collaborator Jing from computer science background developed a method to register each brain slices to the standard Allen Brain Atlas. We generated the first 3D model of the serotonin system, and the locations of all the retrogradely labeled serotonin cells from the 8 injection sites are digitalized and registered to the standard brain so they can be compared fairly. We found that the subcortical projecting DR serotonin neurons mainly locate at the dorsal DR, and the OB and cortical projecting serotonin neurons are mainly located in the ventral DR. And so to visualize the anatomical distribution of projection-defined subsystems, we also established 3D clusters for each subsystem. And here you see that the yellow structure represents DR serotonin neurons. Each cyan structure represents one group of subcortical projecting serotonin neurons, and each red structure represents olfactory bulb and cortical projecting serotonin neurons. I also find that the olfactory bulb and cortical projecting serotonin neurons co-express VGluT3, which is one of the vesicular glutamate transporter. Later, we found that this group of neurons co-release glutamate and serotonin in the prefrontal cortex. So DR serotonin neurons projecting to cortical and subcortical regions are anatomically segregated, but does DR serotonin neurons projecting to one of the cortical regions also send axon branches to other cortical regions? How about the ones that projecting to the subcortical regions? How widely are the serotonin projections collateralized? There could be 2 models. Model 1, serotonin neurons projecting to one region don't project to others. There is no collateralization. And model 2, serotonin projections are highly collateralized. Neurons projecting to one cortical regions also project to others. Same apply to the subcortical projecting ones. To answer this question, I select OFC projecting and CA projecting serotonin neurons. So the OFC projecting ones represent the cortical projecting serotonin neurons, and the CA projecting ones represent the subcortical projecting serotonin neurons. Understanding the collateralization pattern is important because it's like a string-controlled puppet. If the leg and the arm are tied together and then you pull the string, then controls the arm, the leg will also be pulled up. So to label the collateralized branches of serotonin neurons, I injected Cree-dependent flip to either CA or OFC of third mice, so that the expression of flip is only turned on in serotonin neurons that projecting to the injection sites. And then I injected flip-dependent membrane-targeted GFP in the DR to label the projecting fibers. So the whole brain projection pattern is visualized after tissue cleared by a disco and light sheet imaging. So here is a movie showing the 3D whole brain data. The blue fibers are OFC projecting DR serotonin axons. And the green fibers are CA projecting DR serotonin axons. The two subsystems have largely complementary branching patterns. And here is a movie showing that the p-value data of the axon innervation. The results demonstrate that the OFC and CA projecting DR serotonin neurons have largely complementary branching patterns. Based on our whole brain serotonin axon collateralization mapping results, the OFC projecting serotonin subsystem is more biased toward model 2, but the CA projecting serotonin subsystem, in contrast, has less collateralization and is more biased to the model 1. So many known targets of DR serotonin neurons were not innervated by either of these subpopulations, including most of the striatum, thalamus, hypothalamus, somatosensory, and motor cortex. So the DR serotonin system contains at least 2 and likely more than that parallel subsystems. Serotonin system has been proposed as a neuromodulatory system that maintains the homeostasis of brain states. So it is possible that although these parallel subsystems have distinct innervation patterns, they share similar input information. It is also possible that they are more segregated and receive different inputs. To test which hypothesis is correct, I applied the viral genetic tracing strategy developed in my previous postdocs lab, Li Chun's lab, called cTrail, which is short for cell type specific tracing the relationship between input and output. Briefly, I injected Cre-dependent flip to the downstream sites of SERT-Cre mice, so that the expression of flip is only turned on in the serotonin neurons that projecting to the injection site. And then I injected flip-dependent key components for rabies tracing in the DR. Finally, I injected pseudo-rabies virus in the DR to transsynaptically trace each subsystem specific presynaptic patterns. So cTrail revealed that OFC and CA projecting DR serotonin neurons receive biased input from specific nuclei. For example, serotonin neurons projecting to CA mainly receive input from BNST and CA itself. In contrast, serotonin neurons projecting to OFC receive more biased input from LHB lateral hypothalamus. And here is the whole brain statistic analysis showing that the two subsystems receive biased input besides the regions I've mentioned. So the serotonin neurons projecting to OFC also receive inputs from the other medulla nuclei, for example. To summarize, we found that the DR serotonin neurons that project to cortical regions and subcortical regions are anatomically segregated. And the DR serotonin system contains more than 2 parallel subsystems with distinct intervention patterns. The OFC and CA projecting serotonin subsystem receive biased input. So if they receive biased inputs, are they reacting to different stimulations? Do they react differently to the same stimulations? So to answer this question, I applied photophotometry recording to review it. Previous studies have indicated that there is heterogeneity of the DR serotonin neurons responses. The population recording of DR serotonin neurons showed that They were activated when animals receive reward, but not punishment. Later, another group applied optogenetic tagged single unit recording and showed that there are DR-SORT neurons that are activated by both reward and punishment. Meanwhile, there are also neurons that are inhibited by punishment. So could these heterogeneous responses of DR-SORT neurons to reward and punishment result from recordings of different subsystems? To investigate how different subsystems of DR-SORT neurons respond to environmental stimuli, I recorded the population activity of DR-SORT subsystems by applying photophotometry on free-moving animals. So to record the response to reward, thirsty mice were trained to learn the association between the lever press and the sucrose deliver. Every time when as you can see from the movie, every time when the mouse presses the lever, it will receive a fixed amount of water drop. Specifically, I injected Cre-dependent flip into either OFC or CA and then injected flip-dependent GCAM6, which is the calcium indicator, into DR. And then I recorded the population activity while animals performed the task. Here, each line represents an individual mouse, and the time was aligned when the mouse started to lick. I find that both OFC projecting and CA projecting DR-SORT neurons are activated by reward. So to record the punishment-induced responses, I subjected the same set of mice to mild electric foot shock. Remarkably, the two groups of neurons showed opposing responses to foot shock. For DR-SORT neurons projecting to OFC, they are inhibited by the foot shock. But in contrast, DR-SORT neurons projecting to CA are first activated by the foot shock, and later there is a small depression in a subset of these neurons. These findings suggested that OFC and CA projecting DR-SORT neurons receive reward information through similar excitatory inputs, yet only OFC projecting DR-SORT neurons receive reward anticipating action information, but not the CA projecting ones. But they receive largely different inputs that respond to punishment. With so many different anatomical and physiological properties, do the two subsystems modulate the same set of behavior functions? So to answer this question, we applied both of gain of function and loss of function strategies to manipulate these two subsystems and test the behavior phenotypes in a series of behavior paradigms. For gain of function manipulation, we utilized the chemogenetic tools. The HM3DQ is an artificial receptor that can be activated by its agonist CNL. I express HM3DQ specifically in the projection-defined SORT neurons as the same strategy applied for the calcium indicator. Right before the behavior test, the animal receive IP injections of CNL, causing the corresponding SORT subsystems activated. For loss of function, I utilized the TPH2 conditional knockout mice line. I injected Cre to either OFC or CA, and the Cre expression retrogradely knocked out TPH2 from correspondent serotonin neurons. The serotonin is depleted from the correspondent subsystem because of the TPH2 depletion. So here is the summary results of several of the behavior paradigm we tested. To identify serotonin's function, we focused on the results that are consistent between gain of function and loss of function. So these results suggest that CEA projecting DR-SORT neurons promote anxiety-like behavior, but in contrast, OFC projecting serotonin neurons promote an antidepressant effect. So these results provide compelling evidence that the DR serotonin system contains parallel subsystems that are anatomically segregated. The subsystems have distinct and sometimes even opposing functions. And this work demonstrates that the DR serotonin neurons should no longer be viewed as a homogeneous population, and each serotonin pathway's function is specified by its target region. Now we know that the DR serotonin system has different subpopulations. Next, we asked what's the molecular function of molecular foundation of these subsystems. So we used single-cell RNA-seq to profile DR and MR serotonin neurons. To achieve this, we sliced the brain of 14 animals that labeled serotonin neurons with T tomato, and then FACS sort to accumulate a rather pure population. For the first group, we dissected the entire MR and DR area. And for the second group, we only dissect the principal DR without the caudal DR. And after that, we use the SmartSeq to generate the transcriptomic profile. We found that there are 11 clusters in the DR and MR. Each cluster expresses specific molecular markers, including neuropeptides, and GPCRs and different transmitter markers. Of the 11 transcriptomic clusters, so it seems that of these 11 clusters, 6 of them are consistent of neurons from the principal DR, and others are from the MR and caudal DR. So to verify the exact anatomical location of these transcriptomic defined subpopulations, we performed dural in situ labeling with TPH2. And indeed, we find that there are 6 clusters of DR and 1 cluster specified for caudal DR and 4 for MR. So the next step for us is to comparing these data with our previous dissection used by the biogenetic strategy to verify the projection defined and to see whether we can link these different properties, including anatomical, physiological, behavioral, and molecular clusters together. So to achieve this, we needed to develop tools that can actually target the marker genes labeled serotonin neurons, and we have to use more than 2 markers. So we decided to go with the intersectional strategy. In order to achieve intersectional strategy, we made a new mouse line cert flip. Only cert neurons expressed the flip is. We also generated a CRE and flip dural gated MGFP expression retrograde AAV virus to label the axonal projections. So the mechanism for this strategy is only the neurons that express both CRE and flip can be labeled, and only the axonal projections from this kind of dural label neurons will be imaged at a whole brain level. So we chose 2 different populations as example. The first one is VQT3 positive serotonin neurons, and second one is the TRH positive serotonin neurons. We crossed the VQT3 CRE with cert flip and also TRH CRE with cert flip in order to label the 2 different transcriptomic defined cluster of serotonin population. We find that VQT3 positive serotonin neurons are mainly located in the ventral DR, which is consistent with our previous results. If you remember that, we find that the ventral DR contains a large group of population of SART neurons co-express VQT3. And we find that the TRH positive SART neurons mainly locate in the dorsal DR. To quantify the axonal projection pattern in the whole brain of both of these two clusters, we applied tissue clearing strategy and light sheet imaging again. This time we use adipoclear instead of adisco. And you can see that these two populations occupy different brain regions at the whole brain level. And from this movie, it shows that the red fibers represents the axon projections from VQT3 positive neurons, and the blue fibers represents the axon projections from TRH positive neurons. And you can see that these two groups of populations innervate complementary brain regions. And this is also consistent with our previous findings when we use the projection-defined strategy to image the whole brain projection pattern. So next we asked, is there heterogeneity within each of the subpopulation? To answer this question, we collaborate with Professor Qiming Luo and Ming Luo's group, and we applied foremost imaging to reconstruct 50 different individual SART neurons in DR. And here are the examples of them registered in the standard digital brain. So each neurons in different color represent an individual SART neuron in DR projecting to a different brain region. So you can see the ones projecting to olfactory bulb in the meantime collateralized with piriform cortex, while the ones that projecting to the CA collateralized with the ones projecting to BNST, things like that. So to look at these individual serotonin reconstructed serotonin neurons, we analyzed the full 50 neurons dataset. The single serotonin neuron reconstruction results are consistent with our previous bulk tracing data that there are projection-defined parallel subsystem in DR serotonin system. And these data again showed that the serotonin neurons are a very complex system, that there are heterogeneity even within each of the subsystem. For example, you can see that serotonin neurons projecting to amygdala also have different subtypes. Some of them collateralize with BNST, and some of them that do not. And in the meantime, some of them project to the other hemispheres BNST. We also calculated the total axon length of the individual neurons, and we feel a little bit defeated that even the longest neuron in our dataset is shorter than what has been reported for cholinergic neurons in the mouse brain. And here, the longest that we have so far is about 263 millimeters. So to summarize the talk I have just presented to you, we performed a dissection of the DR serotonin system based on the projection pattern. And we find that each serotonin subsystem's function is specified by its target region. And we also find that different properties, including whole projection, brain pattern, cell body location, behavior function, and physiological property are correlated. And later, we applied single cell RNA-seq and analyzed the transcriptomic clusters of the midbrain serotonin neurons, including MR and DR, and dissected the DR and MR serotonin system based on the molecular features. And lastly, we performed single cell reconstruction, revealed that there are heterogeneity even within the projection-defined subsystem. So now we have established the model of the organization of the serotonin system. It is composed of both functionally and anatomically defined subsystems. and the function of each subsystem segregates with anatomical connectivity. But we still are far away from understand this system. And I believe that there are two of the most important questions in the field. What is the role of serotonin in the etiology of psychiatric disorders? And what is the principal function of the brain serotonin system? But to understand the role of serotonin in etiology of psychiatric disorders, we should consider the developmental angle. And that's because most psychiatric disorders emerge during adolescence. The number of previous studies on the topic of serotonin system's very specificity is surprisingly small, and that's partially because the anatomical distinction and functional specificity of the serotonin projections was not clear. And next, it is very difficult to answer the question, what is the principal function of the brain serotonin system? And that is mainly because even though we have some kind of understanding of the organization of the serotonin system, there is a huge gap in between the actual subsystem and their postsynaptic targets. So to close this gap, we need to understand the serotonin synapse better. But 90% of the serotonin synapses are varicosities without junctional specialization, like examples here and here. The use volume release and the postsynaptic targets can be ambiguous. It is not clear how this kind of connection is specified and maintained. So currently, my lab is working on understanding how the system is organized during development. But Rome is not built in one day, and the system is not finished while we were born. So it is built gradually at different developmental stages, and we are using single-cell RNA-seq and whole brain imaging of axonal projections to understand the molecular mechanism of the very specificity and the maintenance of the midbrain serotonin system. Thank you very much for attending this meeting. And lastly, I would like to acknowledge all the my colleagues and co-authors in these two projects. So first, all the colleagues are involved in the two published work from Lichun's lab. I'm very grateful to have the opportunity to work with them, and we are going to build new work based on these two findings. And so my lab in the LMB has almost established. Annabelle Adams is a PhD student, and Jing and Zora are two postdocs, and Robin is a technician. And currently, I'm also looking forward to fully finish the recruitment in my lab and trying to recruit another postdoc. And you are very welcome to apply if you are interested in the topic that I'm talking about. And I'm also very grateful for all the supports that I get from the LMB, especially from the LMB facilities. Thank you very much, and please ask questions and join the discussion.
Thank you for that fascinating talk, Jing, and for introducing us to your research here at Cambridge. Tune in next week when we will welcome Dr. Hatice Gunes from the Department of Computer Science and Technology. Hatice is a reader at the department where she leads the Effective Intelligence and Robotics Lab. Her expertise is in the areas of effective computing and social signal processing, cross-fertilizing research in multimodal interaction, computer vision, signal processing, machine learning, and social robotics. Hatice will be speaking to us on artificial social intelligence from social appropriateness to fairness. Sign up for this talk and others in the series on the links shown below. Follow us on Twitter at CamNeuro to keep up to date with all things neuroscience in Cambridge. See you next time.